Welcome to the Brain and Behavior Research Foundation Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Joseph Taylor will present Imaging-Guided TMS Brain Stimulation Therapy, the right target for the right person. BBRF is the world's largest private funder of mental health research grants supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 190 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic translational and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded $476 million to fund more than 6,800 research grants around the world and across a broad spectrum of brain conditions, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Joseph Taylor. Dr. Taylor is Medical Director of TMS and Director of Clinical Trials and Assistant Program Director and Research Track Co-Director at Brigham Women's Hospital and the Harvard Medical School Psychiatry Residency Program. He's also an Assistant Professor of Psychiatry at Harvard Medical School. Dr. Taylor was the recipient of BBRF's 2025 Claremont Prize for Exceptional Clinical Research and was a 2022 BBRF Young Investigator grantee. Our webinar will begin with his presentation, which will then be followed by Q&A. To submit your questions, please use the question tab on the control panel. Feel free to submit your questions at any time. Following the presentation, I'll ask as many as allotted by the time that we have left. And now I'm pleased to introduce Dr. Taylor. Joe, the floor is yours.
Thank you so much. Can you hear me and can you see my slides?
Yes.
Perfect. Thank you. Thank you so much for the kind introduction. Thank you, Brain and Behavior Research Foundation. This project that I'm going to discuss today would not have been possible without this funding. And thank you to everyone who signed up for the webinar and is here to hear about some of the exciting data that we have. We're talking about personalized brain stimulation today. So why am I showing you a picture of the Sagrada Familia? The Sagrada Familia by Antoni Gaudi changed the landscape of architecture. Gaudi eschewed the latest technology to build it. He hired local artisans to use their hands on upcycled materials. Architecture is very different from science. However, I see a parallel decision with accelerated TMS, transcranial magnetic stimulation, which could change the landscape of clinical practice. Do we eschew the latest technology to build this practice? Do we need our fancy brain imaging? Or can we hire local clinicians to use their hands on upcycled tape measures the way that we do it today? Before I get started, I want to acknowledge the Brain and Behavior Research Foundation funded this trial. I now currently also have funding from the National Institutes of Health, Mass General Brigham's Women's Brain Initiative, and the Home Base Foundation. I have no salary or equity from industry. I do have an in-kind device from Neuronetics for an investigator-initiated trial. And I have no income from intellectual property. We did file a provisional patent on the type of targeting that I'm going to share with everyone today, but there's no income from that targeting. And then acknowledgements up front as well. Mike Fox and Sean Siddiqui, both BBRF supported investigators, helped with this trial as co-senior authors. I want to acknowledge the physicians who cover our TMS clinic. They allow me to do the research and to be here with you. And then all of the residents, fellows, research assistants, clinical research coordinators who day in and day out work with our study participants and our patients and the data that we collect. A tremendous thank you to all of them. And then finally, to get a little serious here, you're looking at a picture of Dr. Mark George, who's largely credited with developing clinical elements of transcranial magnetic stimulation on the far left. The late Nolan Williams pictured in the middle here, and then a picture of me with the two of them. This picture was taken at the Brain-Stimulation conference in Japan last year. Sadly, as many of you know, Nolan Williams died by suicide 8 months later. He was a good friend of mine. We had known each other for about 15 years. And I've started to show this picture at the beginning of every talk to try to destigmatize depression and suicidal thoughts. If the inventor of something that pushed the field forward so far can struggle with depression, then anyone can. So please, if you're struggling, get help. Some learning objectives here. I want to keep this very straightforward in case there's people on the call who aren't scientists or physicians. I want you to be able to describe conventional TMS for depression, describe accelerated TMS for depression, and then explain the role of imaging in accelerated TMS or ATMS. So those are our basic goals, and I hope to be able to convey those to you. The need is a better and faster treatment for depression, but also across the diagnostic categories. And what you're looking at here is the outside facade of the Sagrada Familia. And actually, this facade was created at a time when Gaudi had a period of depression in his life. And interestingly, you can see the depression manifest itself in the architecture. This side is much more angular. It's much more moody. If you look at the figures in the bottom corner, you can see basically what Gaudi was experiencing in the architecture. The intervention is transcranial magnetic stimulation. And for those of you that aren't familiar with this is a focal external form of brain stimulation. It is non-invasive. It uses basic principles of electromagnetic induction. It basically provides us with a way to drive brain activity. So this isn't some magic wand. All it does is increase or decrease brain activity based on where, when, and how we stimulate. We have FDA clearances that are coming through very rapidly. Most of what we use TMS for clinically is depression because that's the widest insurance coverage that we have. But it also has FDA clearances for anxious depression, migraine headaches, OCD, smoking, and most recently, within the last couple of weeks, post-traumatic stress disorder or PTSD. There's a breakthrough device designation for bipolar depression, but we don't know yet how to use it effectively. There's been other BBRF funded investigators like Yvette Shaleen who've done amazing work in this area. But this is a rapidly evolving area, and we're just beginning to understand how to use this tool. And just to reinforce the point, we know that conventional TMS works. I'm going to talk a little bit about the limitations of conventional TMS and why we're moving to something like accelerated TMS. But just for everyone's background, this is a meta-analysis published this year on conventional once-daily TMS, which we use to treat depression in the clinic. They looked across the literature and then basically analyzed the results. Without getting too far into the details or the complexity of the statistics, the green box here just highlights that the odds of getting better with active TMS versus sham is 4 to 5 times. And you're 3 to 4 times more likely to remit. In other words, feel totally well once you get TMS, which is widely available and insurance covered. And interestingly, they also found that the dropout rate between active and sham TMS was no different. So this also reinforces the idea that this treatment is, generally speaking, very well tolerated, and it's pretty effective. We have head-to-head trials showing that conventional once-daily TMS, the way that we do it now, outperforms the next medication that you might try. And these 2 papers are published around the same time in 2 very prominent journals documenting that TMS outperforms a medication switch or a medication augmentation. So really good high-quality evidence that the conventional way that we do TMS works. We published this article looking at resource utilization because we understand that the complexity of the healthcare system involves cost. So we looked at Medicare data for TMS, And to keep this simple and quick, the take-home messages from this analysis was that TMS, just conventional once-daily TMS, saves Medicare money, that the people who get TMS use the system less often and end up using outpatient resources rather than emergency or inpatient resources. So it actually saves money. Unfortunately, it's still widely underutilized. Less than 1% or fewer than 1% of people who were eligible for TMS in this data set actually ended up getting it. So kind of a public service announcement here, conventional TMS, the way we do it now, works and it saves money and it's still widely underutilized. So probably a separate conversation. However, we're here to improve the treatments that we have now. And there are limitations to current versions of TMS for depression. The first is that it is a daily weekday treatment for 6 to 8 weeks. So it requires you to come in every single day. If you started TMS today for depression, you would have to come in 5 days a week, and you would be through the end of August into the first day of September before your treatment course was ended. So that's a huge time burden. Next, there's target uncertainty. I took this screenshot to show you that I'm in Boston, but essentially we use scalp-based measurements to target the conventional form of TMS right now. So we don't do brain imaging. We take a scalp landmark approach that is inexpensive and quick to implement. So while you can see that I'm in Boston based on this screenshot, there's much more sophisticated ways to look precisely where I am in Boston. And so this radius just gives you a very broad picture. And that's sort of how I think about scalp-based measurements. They're probabilistic, but we really don't have a way to peek into the brain and see exactly what we're targeting. And that's the core of this talk today. And then finally, conventional TMS for depression, the response rates are about 50, 55%. Some argue that it's up to 60, depending on the type of study and how you look at it, whether it's open label or not. But in general, it's right around half of people get 50% better. So we can improve TMS. We can improve the time burden, we can improve the targeting, and we can see if those two changes translate into more people feeling better. So the intervention I want to talk with you about today is called accelerated TMS. And this was largely pioneered by Nolan Williams. But there were people long before Nolan who had started to develop this idea, dating back to 2007 when Colleen Liu tried basically giving 2 TMS treatments in a day versus 1. So the broad definition of accelerated TMS is at least 2 treatments per day rather than just 1. So these are landmark studies that investigated whether you could give multiple treatments in a single day to speed up the effects of TMS. Paul Holtzheimer published this paper in 2010, and then Mark George published this paper in 2014, looking at multiple treatments per day. All of these led up to, and this isn't all of them, Nolan Williams's seminal letter to the editor in Brain in 2018, where he developed what he called high-dose spaced beta burst TMS. Now, he recruited people who would otherwise have tried everything that we have, ketamine, S-ketamine, dozens of medications, dozens of forms of psychotherapy. These people, electroconvulsive therapy too, these people were basically considering deep brain stimulation or neurosurgery for depression. And Nolan and his team here took an educated guess on how to get them better faster, and it worked. They followed up with this amazing publication that was an open label study in basically 21 people who had severe depression. And this study showed that if they gave them, for the first time, what they called SAINT, Stanford Accelerated Intelligent Neuromodulation Therapy, that the remission rate, so people who felt totally well, 90.5% of them remitted with this treatment. They felt totally well. Those numbers are really hard to find in psychiatry. They rival electroconvulsive therapy, ECT, which has been our best treatment for about a century. So this created a lot of buzz in 2020, but there was still a lot of skepticism that this was real because it was open label. They published a double-blind trial in 2022 as a follow-up. In this study, they basically gave that same SAINT treatment. They now called it SNT for SAINT to keep it shorter and simpler. They basically treated 29 people with either active or sham SAINT. And at the planned interim analysis, they found that the MADRS score, which is a clinician-rated depression scale, diminished by 52.5% in people who got active versus 11.1% in people who got sham. So this was the double-blind data that the FDA needed to issue a clearance of this entire Saint package, which is a hardware system and a target. We'll break out what the pieces are of this intervention. But this was a huge step forward in the field. The data suggests here that the MADRS score drops within a single week. So the active treatment arm is in gray and the sham treatment arm is in dark gray. You can see these groups separate within a single week of treatment. And then that persists out 5 to 6 weeks. It doesn't matter if you use a different clinician rated scale, you still see a nice, clean, clear separation. And the authors of this study ended up shutting down the trial because they could not justify giving people sham treatment when they had such a clear separation with active. Now, the authors of this SAINT protocol, Cole et al., which is how I'll generally try to refer to this protocol, since now SAINT is a trademarked and intervention tied to a company called Magnus Medical that are trying to bring this to market. So what did Cole et al. Do that really differs from what we do with conventional treatment in the clinic? Well, they changed at least 5 different variables: the schedule, the dose, the intensity, the targeting, and the precision. So they gave 10 treatments per day for 5 consecutive days instead of 1 treatment per day for 30 to 36 weekdays. They also basically tripled the dose of each treatment. So if you go into the clinic and you get what we call theta burst stimulation, it's only 3 minutes long. They gave 9 minutes of theta burst each time they treated. They changed the intensity. So in the clinic, we usually use 120% of motor threshold. Cole et al. Used a lower intensity, but they adjusted it for what we call scalp to cortex distance. That metric can only be done if you have the person's brain scan. They also used MRI-based targeting with a patented algorithm versus a scalp-based targeting metric that we use in the clinic. Again, those head measurements. And then finally, they use something called neuronavigation, which is basically just a fancy camera system that tells them precisely where the TMS coil is targeting the brain, versus in the clinic where we basically use visual inspection alone. So as you can see, they changed at least 5 variables at once. The scientific method usually asks us to hold everything constant and change 1 variable so that we can understand the impact of that variable on the outcome. When I talked with Nolan about this when he was developing SAINT in this high dose space theta burst, I think his goal was to try to take a big swing. Let's optimize everything we think we can do with TMS all at once, acknowledge that we don't know which this recipe is important, see if it works, and then work it out from there. And obviously that swing was successful. We now have a treatment that is likely to be covered by insurance this calendar year. At least some insurances will be covering accelerated TMS this calendar year. But now the field is left trying to figure out which parts of this recipe are important. And the one that caught my attention the most was this MRI-based targeting. Now, I don't have time to go through the targeting history of transcranial magnetic stimulation, but quickly, and Robin Cash wrote a really good review of this, where we started TMS by just moving the coil 5 centimeters forward from the part of the brain that used to make your finger twitch, called the motor hotspot. This was just a very probabilistic way to identify the treatment target. Then we got a little fancier. We started using the 1020 EEG system from neurology, and we started going to an F3 spot. Will Beam is the name of an RA who developed this and put it out publicly and freely available. So some clinics do use the Beam F3 method instead of just moving this fixed amount of distance. Then we tried anatomical targeting. We eventually moved to functional imaging like PET to try to target TMS. Then we moved to functional connectivity targeting. Now you can do this at the group level where you can just neuronavigate to a specific coordinate in the brain that works for a bunch of people and then stimulate there and see if the outcomes get better. But what the field really is interested in most right now is personalizing this. Can we scan each individual patient and then look at some pattern of activity in that brain of that patient and then pick the target based on that activity? So what's the big deal? Why don't we just adopt imaging and try this, since we have the tools and we have some signal that imaging might improve outcomes? Well, it's about scalability and cost and non-specific effects, right? Not every place has access to an MRI scanner. Not every place has access to servers that can process the data. Not every place has access to the neuronavigation equipment that you need to buy and and largely master to be able to deliver the treatment the way that Cole et al. Did in their seminal papers. All of these things require space and cost and expertise as well. Selecting a brain circuit target for TMS is quite complicated, and the neuroimaging experts that are computational neuroimagers are often not the same people who are doing patient-facing work in the clinic. So this isn't as simple as just adopting it because we think it's going to help. But I'm also thinking about Anthony Gaudi, right? And his message here. And this is just a cartoon from the oatmeal suggesting that all of us tend to get distracted with shiny new things. And there's always something new with imaging or some tool that we can add on to TMS to make it seem like it's going to work better. But I think it's worthwhile for the field to take a minute and run some of these studies to make sure that we're not just getting distracted, that these tools actually help people. Now, taking a step back, why would we think that imaging would improve outcomes? Well, I'm showing you a picture of a tape measure because this is how we target TMS in the clinic and how we have targeted for 15 years. Again, we have meta-analyses showing that this method works and it works well. But how could we not be able to beat a tape measure? Tape measures are used to tailor clothing. Using it to guide brain stimulation on its face seems like it's hard to believe. What are the reasons for doubt in using this old internet meme? Well, there's a lot of reasons to suspect that imaging may not actually improve clinical outcomes for TMS. We have 2 decades of scalp-based targeting. We know that this works in randomized trials and in naturalistic clinical settings. Maybe there's a ceiling effect for TMS. How good could it be? We're already outperforming medications and therapy. How much better could it be? Well, maybe it's spatial precision and target overlap. Maybe the TMS tool is just too crude to have millimeters actually matter in imaging. We also know from other data that more treatment tends to correlate with better outcomes. So if you're looking at what changed in the Cole et al. Protocol, the SNT protocol, most people that I've talked to, and myself included, would guess that it's the increase in the number of pulses that they give and the consolidated treatment schedule that matters the most as the single most important thing that would lead to better outcomes, faster and better outcomes, not imaging targeting. We have some reproducibility concerns in imaging. We've known for a couple of years now that the way that we've done imaging for years wasn't really the best way to do it, and that there's some reproducibility concerns here. We have some data retrospectively that if you target someone close to their imaging-based target, just on accident with their scalp-based landmarks, those people tend to do better. But that's not the gold standard for the field. That's retrospective data. That's looking at the outcome after the treatment's already happened. What we need to do is have prospective data where a group of people get imaging-guided TMS and another group gets scalp-targeted TMS, and we compare the outcomes. I'm not the first person to say that. There have been plenty of studies that have tried this in the past, but we haven't really seen a lot of good results. There's a couple papers I want to highlight here. The one on the left is basically what was published in Brain-Stimulation in 2024, where they did what they called a pragmatic TMS, accelerated TMS protocol. So they basically took an insurance-covered TMS session, so 36 TMS sessions, and they made their own accelerated treatment protocol. Instead of 10 treatments a day for 5 days, they did 6, 8, 8, 8, 6. The other thing is that they didn't do any fancy imaging. They didn't use any fancy neuronavigation equipment, no scalp to cortex adjustment. They just gave TMS like they would if you walked into any average clinic in the country. They just gave the different pulse numbers per day. And they found really good results, better than conventional TMS. Not quite as good as Cole et al., but it suggests that maybe imaging isn't that important because you can do it with scalp-based landmarks and no fancy brain scans, and we're getting better results. Then we have this study on the right, published in Nature Medicine, that looked at connectivity versus scalp-based daily TMS and found no difference. Now, this was a huge deal when it was published because it was in basically 250 people. The problem is that this protocol deviated from conventional TMS in several ways. They only looked at 20 sessions instead of 36. The targeting method that they used is not the one that the Cole et al. Protocol would suggest is best. and it doesn't particularly align with what the rest of the field would suggest, because I believe they were targeting the insula as opposed to a region of the left dorsolateral prefrontal cortex that links to the subgenual cingulate. So we can't really reach conclusions on this, but this was yet another reason for skepticism that any of these fancy brain scans improve outcomes. So we set out here to test this hypothesis with accelerated TMS. We wanted to match what Cole et al. Did as closely as we could and do that version of accelerated TMS with connectivity-based targeting and without connectivity-based targeting in a blinded way. The challenge is that, as I mentioned, the targeting algorithm that was used by Cole et al. In the SNT paper is trademarked. Now, we know what it is because the field for a very long time, people like Helen Mayberg and others, have been arguing that it's the subgenual cingulate cortex, which is a region we can't reach directly with TMS, but we can reach it indirectly if we hit the right target node on the cortex surface. So we know in general how they use their targeting, but at the time, because this was being developed, my friends over there couldn't share the algorithm. So I had an option. I could either try to replicate their algorithm, or I could come up with our own algorithm. And I work in the Center for Brain Circuit Therapeutics with wonderful colleagues like Mike Fox and Sean Siddiqui. So we decided to use our own algorithm instead. And this sort of reminds me of the internet joke that people have, where if you grew up with not a lot of money, and that includes me, and you wanted a Dr. Pepper, you might ask for it at a restaurant. And your parents or your guardians might say, no, you can't get Dr. Pepper. That's too expensive. We have Dr. Pepper at home. And the Dr. Pepper at home is actually Dr. Perky from Food Lion. That is a real product, by the way. So in this study, we did not use the patented SNT or SAINT algorithm from Magnus Medical. We used our own internal targeting. I will explain it, but it is quite complicated. But I want to just make that point. And again, the reason we're doing this is that imaging adds cost and complexity to accelerated TMS. And also, we don't really know how to interpret the signals from imaging. We're so used to putting people in the MRI scanner and then seeing what lights up. But if you're using those regions that light up to guide your brain stimulation, you really need to have an idea of what those signals actually mean. Are they a cause of the illness that you're studying? Are they a compensation for the illness that you're studying? Are they an epiphenomenon altogether unrelated to the illness? Are they showing that that person got treatment rather than the illness itself? So we need causal information from imaging, and we usually can't get it with conventional imaging ways. And then finally, there's reproducibility concerns. So how do we get around these? How do we do a better job of targeting? Well, there's different ways to do it, but we do know that you need hundreds or thousands of people to have a brain-wide association signal. And again, that's not how we've been using imaging for a long time. So you can build up larger data sets across sites, or you can use meta-analyses to boost your confidence that your imaging signal is something that is reproducible, that you can interpret and use to guide brain stimulation. So the way that we think about targeting is through something called network mapping. So Mike Fox is largely credited with pioneering this work. The review article is shown here in the New England Journal of Medicine. Basically, the idea here is that we work backwards and we try to take advantage of serendipity. So when someone has a brain lesion or a brain injury from a focal damage or a stroke, we have unique insights into that unfortunate event, right? Because that area of damage might cause a specific symptom to happen after that area is damaged. That is a causal insight into the neuroanatomy, and that's exactly what we need to do. So to give you an example, this is a review by Sean Siddiqui. If you take 6 different strokes that afterwards resulted in depression, you could try to look and see where these lesions all overlap in space and say, aha, this red area, when damaged, results in depression. Maybe that means that we can stimulate that area and relieve depression. The problem with this rationale is that, as you can see, not every lesion that resulted in depression overlaps in one space. How do you explain this lesion or this lesion or this lesion? You might incorrectly assume then that these lesions don't converge onto a same circuit. Our network mapping approach takes a different idea. So instead of looking at the lesions and expecting them to all converge into one single area of the brain, we ask a different question using the human connectome, which is basically a giant wiring diagram of the human brain. So we ask, Are all of these lesions connected to regions in common? And do they go up or down in activity in a synchronous manner? And if you take that question and you look at each lesion and its connectivity profile using the human connectome, you do see that even though these lesions don't overlap in a single area, they do overlap and converge in a same circuit. So all of these areas work in concert with one another. And then that target then becomes a whole brain circuit that you can use to stimulate for TMS. So that's how we did the targeting here. This lesion network mapping technique has been used in so many different ways across symptoms and functions to try to localize the functional neuroanatomy of everything from aphasia and hemichorea to tics to pain to criminality and even addiction remission. So we have a rich data set here of brain lesions that resulted in focal damage and an onset of a specific symptom or condition. And that's how we in the Center for Brain Circuit Therapeutics think about developing TMS targets. And we've written commentaries about this. I sort of think of imaging and psychiatry as telling us where to go, not what we're doing there. For years, we've hoped that imaging can help us develop biotypes of depression or track someone's progress through an antidepressant treatment. But those studies have not really worked well for a whole host of reasons. So instead, I think we're becoming confident that imaging can't tell us what we're doing there, but it can tell us where to go. And in that way, it's sort of like a surgical model, right? If you have a tumor and you take a brain scan, that scan doesn't tell the surgeon what type of tumor it is. That's tissue pathology. But it does tell the surgeon where they need to go and what structures they need to avoid. And I think that's sort of how imaging might end up having a role in psychiatry. The depression circuit that we targeted in this study was derived from a study by Sean Siddiqui and Mike Fox published here in the Nature of Human Behavior journal, where basically they looked across 14 independent data sets. So they looked at brain lesions that were associated with post-stroke depression across these different sites. Then they looked at TMS coordinates that were associated with depression relief. And then they looked at deep brain stimulation coordinates that were associated with depression relief. So across these 14 independent causally informed data sets, the question became, do all of these things connect to the same circuit? And is it a depression circuit that is sensitive and specific to depression? That when damaged, this circuit reliably causes depression? And the short answer is yes. If you look at the connectivity profile of brain lesions that were associated with depression onset, and TMS sites that relieve depression and DBS sites that relieve depression, they look like the same map. And there's a lot of fancy statistics that were run to confirm that this map was sensitive and specific to depression and not a bunch of other symptoms or syndromes that may have also been there. So this causally informed convergent depression circuit was the target for this trial. So we had a very simple pilot study goal. Number one, we wanted to emulate the Cole parameters and protocol as closely as possible, and really was so kind working with Nolan and his group who were supportive of this. Next, we wanted to just isolate the variable of imaging, of connectivity-based targeting. So everything else about these two groups was identical. The only difference was that one was going to get treatment at their connectivity-based target of the convergent depression circuit, and the other people were only going to get treatment at their scalp-based targeting. But it's a triple-blind study, so no one's going to know which group they're in. And then finally, we just wanted to calculate the effect size of connectivity-based targeting. We did not know coming into this study if we would be powered to detect a difference because no one had done this specific study with accelerated TMS before. So we just cautiously said our goal is to measure an effect size. How much of an effect would connectivity-based targeting have on accelerated TMS? So that was the study in a nutshell. I want to make sure we highlight this. We matched Cole et al. On everything in these 40 individuals who had depression. So 10 treatments a day for 5 days, 1,800 pulses per treatment, 90% motor threshold adjusted for scalp to cortex distance. The only difference is in our study, one group got connectivity-based targeting and the other group got scalp-based targeting. Everything else was matched line by line. Now, you could say, well, why didn't you do something different? Why didn't you just do this as a pragmatic protocol and compare it to the Cole et al.? Again, I wanted a single variable, and I wanted that variable to be imaging-based targeting. That's the only difference between the groups. So here is a methods figure, and this paper is published now. So we took each participant, we took a brain scan from everybody, and we calculated their connectivity-based target and their scout-based target. We did that for 40 people that we enrolled. Everyone had a connectivity-based target and a scout-based target, and then we randomly assigned them to one of those two targets. That's the study. How did we do this targeting? I'm going to gloss over this in the interest of time because I know this is also complicated and we have probably a broad audience. But basically, we took this whole convergent depression circuit. We looked at every single voxel in the brain and looked at the time series of every single voxel in the brain and created a weighted mean of the whole brain. Next, we had a search space called the left dorsolateral prefrontal cortex. And within that search space, we modeled every single possible TMS target that you could stimulate within that search space. And we derived an average time series from every single TMS model. And then we picked the TMS target that looked the most like that whole brain connectivity. So which region in that left dorsolateral prefrontal cortex had the strongest connection or the greatest time course correlation to that whole causally informed depression circuit. That's how we did the targeting. And if anyone's seen It's Always Sunny in Philadelphia, this is how I feel every single time I tell someone about this targeting approach, because I understand that it's very complicated. But our goal is to try to simplify this. And if we validate the process, we will come up with a way to make it easier to understand and easier to use. So here's the consort diagram. We assessed 211 people for eligibility, excluded 151 because of inclusion-exclusion criteria or people who were lost to follow-up. We screened 60 people formally, 20 people were excluded, and then we randomized 40 people, 20 in the connectivity-based targeting group, 20 in the scout-based targeting group, and then we analyzed the results here. One person discontinued from the scout-based targeting group because they withdrew after 30 of 50 treatments. We did include their data in the end, which is why we counted 20 versus 20. This person anecdotally got 3 days of treatment and felt so well that they wanted to go back to work. Here are the demographics from the study. The population was slightly biased toward women. Age on average was about 45. You can see the ethnic and racial self-identification statistics here. These people had substantial depression. They were allowed to have general anxiety and they were allowed to have ADHD, but largely no other diagnoses. And again, this was based on Cole et al. We've done some other clinical trials to start to begin to explore whether we need to adjust the targeting based on comorbidities. The baseline score for people coming into this trial, the MADRS was about 32.5, so right on the high end of moderate, close to severe. Maudsley staging method is a way for us to quantify how treatment resistant the illness has been for them based on the number of medications they've tried, whether they've had augmentation, whether they've had ECT, and the duration of the illness. In general, everyone in this trial had severe treatment resistance. And on average, people came in with about an antidepressant trial number of about just over 5 medications that they've tried, in addition to psychotherapy. We let people stay on their medications, but we did ask the medications to be consistent and stable as people went through the trial because we wanted to attribute any main effects or side effects to the intervention rather than any variables changing in the background. This is the primary outcome. So we're looking at change in MADRS, where lower or greater change would signify greater improvement between scout-based targeting in green and connectivity-based targeting in purple. So you're looking here at baseline. Everyone starts at 0, and that was normalized at baseline between the groups as an a priori statistical decision consistent with Cole et al. So everyone starts at 0. And then day 5 of treatment, connectivity and scout-based targeting completely overlap one another. Basically, everyone, on average, not everyone, but in general, both groups got better, substantially better. So within 5 days, remarkable results. I was stunned to see this in person when we first started doing it. 1 week out, you start to see a little bit of a separation, but the groups overlap. 1 month out, if you look at the MADRS change, you see a between group difference and a clear separation. Again, this was blinded, so the participants didn't know which group they were in, the technicians didn't know which group they were in, and the clinician rater didn't know which group anyone was in 1 month out when these scores were collected. But the response rates differed from 80% to 60%, and remission rates were 65 versus 50. And the median MADRS reduction was 83% versus 62%. The main effect size of targeting was 0.8. That is a large effect size, and that was what we sought to measure in this study. This difference was statistically significant, but again, our primary outcome was really the effect size. And remember, this is not an effect size of treatment. This is an effect size of connectivity-based targeting alone. Now, it doesn't work for everyone. You can see some of these people up here in the connectivity group did not get substantially better, but on average, if you were in the connectivity-based group, you had better odds of feeling better and having a greater change in your depression rating scale. I still have not acquired this picture, but Sean Siddiqui, who helped me with this trial, took a screenshot of my face when we unblinded these data because I was stunned. Cynically, I came into this trial thinking that scalp and connectivity-based targeting would not matter, and that I would have a hard time getting this study published because I was also skeptical, despite my work in the area, that prospectively we would see a difference, especially in a sample size like this. So I was stunned to see this result. And I think this is some of the first data in psychiatry to show prospectively that imaging improves outcomes. Now, there's a bunch of caveats to keep in mind here, right? One of the big questions has always been, well, do we actually need the scan? Is there some adjustment to the scalp-based measurements that we already do that can substitute for the scan? So we mapped each target, and you can see looks like there's a little bit of a separation where maybe the connectivity-based group got treatment a little more lateral than the scalp-based group. And that is true if you look at the coordinates themselves. But if you include these coordinates in the statistical model, it does not explain the difference between the groups. Now, if that's hard to understand, just take a look at this heat map. The warm colors are the people who did the best in the trial. The cool colors are the people who did the worst in the trial. If this was simply a function of moving lateral, then you would see all the warm colors clustered on the lateral side. In reality, you see a mix of colors, suggesting that it's not just a spatial location that is correlating with the result, and that maybe there's still some sort of signal from the connectivity that you couldn't get from the scalp-based landmarks alone. The other thing that we addressed in this study, which rarely is addressed in other trials, is are these targets reproducible within person and different between people, right? If you're arguing that these targets are actually personalized and useful clinically, then they would have to meet both of those criteria. They'd have to be reproducible within person and different between people. So what we did was called a split half analysis. So to get the target for each person, we collected 40 minutes of resting state data, multi-echo resting state data. That is a lot of data, and no study has ever picked a target with that much data prospectively, to my knowledge. But that 40 minutes of data let us split the data into 20 minutes and 20 minutes. So this was before anyone had TMS. We collected their 40 minutes of data and we split them into two 20-minute intervals. And then we calculated the target in each one. And that allowed us to look at the difference to say, if you split this data in half and you calculate target 1 and target 2, how much does it move? And then you can compare targets between people to test your hypothesis that they're different between people. So we have an intra-individual difference and an inter-individual difference. And what we found was that if you look intra-individual, so if you take the target and you calculate it twice before someone gets treatment, the median difference is only 4.4 millimeters. Compare that to the distance between people, where the targets separate by about 12 to 13 millimeters. And that was a statistically significant finding. So we tried to argue, really, that this shows that the target doesn't change much within person before you stimulate, but between people, the targets are quite different. Again, further reinforcing the idea that maybe we're getting something important from imaging. Now, again, before we run off and conclude that this is, a solved question, we did not find a between-group difference with anxiety ratings, shown here. And we did not see a difference in a different depression scale, which was not our primary outcome, called the Beck Depression Inventory. This is a self-reported scale. You do see the characteristic separation here. It just didn't separate enough to reach statistical significance. We are collecting the long-term data now. We should have that analyzed soon. Unfortunately, it's still the BDI, but this did not separate significantly in the way that it did for the MADRS. And I'm happy to kind of unpack that and postulate as to why we saw it in one score but not the other. But the MADRS was our primary outcome measure, and it's a clinician-rated scale that's consistent with the other kind of accelerated protocols. There were no side effects between the groups that were different that would have destroyed the blind, and we confirmed that the blind was still intact. But basically, all of the side effects that you would expect to see with TMS, we saw in this study. There was no difference between the groups in terms of side effects. So what have I shown you? The median MADRS reduction with connectivity-based targeting was 24 versus 18 in the scout-based group. That was a statistically significant difference. The effect size, the Cohen's d measurement, was 0.8. Again, that's a large effect size. Most of our treatments in psychiatry don't have that large of an effect size, and this is just something that we added on, right? This is just targeting alone, not the effect size of treatment. And then finally, we use something called the number needed to scan. I'm guessing someone somewhere had thought about this before us, but I didn't find a lot of literature on this technique. So the number needed to treat is an established metric across medicine that basically gives you insight into the clinical impact of what you're doing. Here, we turned it from the number needed to treat into the number needed to scan. And in our study, you had to scan 5 people to get one single person better at that 1-month mark. That is a very low number. We use a lot of different interventions in medicine with much larger number needed to treat numbers than 5. So again, all of this was quite surprising to me. How do our results compare across studies? Well, we showed that with connectivity-based targeting, we had an 83% improvement. Response rate was 80, remission rate was 65. That is consistent with the Cole et al. Open label studies, the Cole et al. Randomized trial, and the Lure et al. Pragmatic protocol that didn't use connectivity-based targeting but used scout-based targeting. So in general, all of these things are starting to align. If you use connectivity-based targeting, it seems to result in better outcomes. And our studies, I think, the first to show prospectively that it beats scout-based targeting. But these numbers are also reassuring just for accelerated TMS in general. Building a literature here to show that the response rates with accelerated are better. And our studies, I think, one of the first to show that maybe those actually get even better if you use imaging. So why is this important? This isn't just an academic exercise, right? There are now T codes listed here for personalized connectivity-guided accelerated TMS. Blue Cross of Mass and Rhode Island, and probably other areas as well, have agreed to reimburse this clinically this calendar year. So we are starting to gear up for the possibility of an accelerated TMS launch. This could really change what the landscape of TMS looks like. So it's important to know if this connectivity part actually matters. Now, there's been a lot of developments in the field lately. The BrainSway company with their H-coil got their own clearance that doesn't use or reference accelerated, but that's not translating. So there's a lot of nuance here. But if you're using these T codes, then I think you do have to use connectivity guidance. So it's important to know if that matters. As I said, our trial was one of the first ones to test this and show it prospectively. There is a preprint from this group in Acacia Clinics that have been doing accelerated TMS for years. And they did scout versus connectivity based, and look that they found the results that look very similar to ours, with a 77 response rate for connectivity versus the 66% response rate for scout. Now, this is open label. It's not a randomized trial, but they used the same targeting that we did because Sean Siddiqui, who's the senior author on this, was actually the person that helped them pick their targets. And in this data set, connectivity-based guidance was the only independent predictor of response. So a really compelling alliance. Now, again, this is just a preprint, but I was reassured that our results look pretty close. Why did we show that targeting mattered in a way when other studies did not? Well, I think we were the first ones to replicate the Cole et al. Accelerated protocol, which probably has a bigger effect size. But also, I think it has to do with what we talked about earlier, which is we think about causally informed targets at the whole brain circuit. We use 42 minutes of data, multi-echo data. That's 16 times what most studies use. So I think we might be undersampling when we're picking targets otherwise. We're targeting a whole circuit, not just a brain region, and we think that that matters. And then finally, we think this is causal convergent evidence across other brain interventions that treat the same illness. Now, this number needed to treat, I'm mindful of the time. basically, this metric that we found is really compelling. So in order to prevent one death from breast cancer, you have to scan 368 people with mammography. To prevent one death from lung cancer, you have to screen 130 people with a CT. In first episode psychosis, which is, I think, the only guideline-based use of imaging in psychiatry in the clinic, you have to scan 18 people to have something that's potentially clinically actionable. Doesn't explain the psychosis necessarily. There's just something there that might be clinically actionable. 18 people. And this is part of the APA guidelines. There's really no other part of psychiatry where we routinely scan people. The number needed to treat in the study or scan was 5. Now, this isn't an apples to apples comparison, right? Because these are screening for death, and this is a screening for some other actionable item versus something that is optimizing treatment. But it does put it into context, and I had a hard time finding other examples of studies that did what we did, which is test whether imaging actually improves outcomes. And I think the reason for that is that in other parts of medicine, and I'll kind of counter argue myself here, in other parts of medicine, it's extremely rare for them to say, hey, we have this technology, but we don't have double-blind data showing that it improves outcomes, so we're not going to invest in it, right? You would never do a PCI, you would never deliver radiation, you would never even do a needle biopsy without imaging if you had access to it. So I do think, while we're trying to be mindful about scalability and cost, the counter argument to that is no other branch of medicine holds themselves to this high of a standard. They spare no expense for the illnesses that they treat. And it's really rare to see double-blind data with and without imaging. So in psychiatry, I think there is a risk for us to be holding out for even more data and even more data to convince ourselves that imaging matters. In all likelihood, it doesn't hurt. Does it help? Our data suggests that it does. I understand the desire to have even more data before we widely adopt this. But again, the risk is that our evidentiary bar gets so high that we don't implement this. And depression is something that can kill people. There's a lot of limitations to this study, right? It's a single site, small sample size, although I will say that this trial was bigger than the SNT paper that got FDA clearance, right? This is, to my knowledge, the largest study that used this specific protocol. And I know that's rapidly changing and people use different parameters, but keep in mind, 40 people with accelerated TMS, each person got 50 treatments. This is a lot of work. So we're going to have a lot of challenges scaling this up to get trials with hundreds of people. There's no sham, right? But we beat out an active treatment, and that's compelling. And we had a 1-month primary outpoint. I didn't ask people coming in what their biases were. Anecdotally, everyone wanted imaging. So we can do a better job of assessing that. And then generalizability, right? We use really narrow imaging criteria, and I won't go through this in the interest of time, but suffice it to say that we only sampled people who had depression and anxiety because that's what Colitol did. And it's possible that different people need different targets based on their comorbidity profiles. And then finally, we just used a lot of data to pick our targets, and our targets were complicated. So obviously, those are all limitations. Back to the Sagrada Familia, I wrote this paper and basically did this presentation expecting to have a null result and saying, hey, look, we did this major accomplishment without technology. Maybe we don't actually need the technology for accelerated TMS and these fancy brain scans. And then the results were the opposite of what I expected. So I think the twist on this story is that while Gaudi did design this without the latest technology, he never finished it, right? And now today, we know that the Sagrada Familia is finished, and they certainly use the latest and greatest technology to finish this masterpiece that was started without the technology. And then the final twist for you is that I was flying back from that Japan meeting, and I saw this article in the New York Times that Gaudi is considering they're considering Gaudi for sainthood, which is a nice play on words with the saint and brings us full circle. So I will stop there. I apologize for going a little long. Thank you so much for your time.
Joe, thank you for an outstanding presentation. And really going into detail, you answered a lot of the questions that came in. So we have time for one question. I want to ask you, where do you see this going if we were to have you come back in 3 years, 5 years, 10 years? What would you be telling us?
Yeah, that's a great question. So right now, I'm hopeful that the R01 grant that I wrote to have a confirmatory efficacy trial gets funded, because I would like to see this in a larger sample size. But I think that the insurance-based coverage of connectivity-based targeting has really gotten ahead of the data. Um, because they basically agreed to this before we even had our results, but that's because Colidol did such a nice job of getting it through the process. So I think we will start to see accelerated TMS grow throughout the country and hopefully the world. I think we will start to see more imaging-based targeting according to the principles that all the pioneers before us came up with in terms of targeting. I think you will see our group start to test different brain circuit targeting techniques, just like we did for depression with other indications. We currently have trials going for perinatal depression, post-traumatic stress disorder, and smoking, where we're testing, again, this idea of imaging and whether it actually improves outcomes in any tangible way. So I think that's where you're going to see the field going. And then finally, we're going to need to see if this works well in people who have comorbidities. And we're going to have to test new targeting techniques for people who have comorbidities. The criticism from science in general is that while we have the purity of the randomized trial, and we can control all the variables, when you bring it into the real world where people have comorbidities, they have medications, they may not have met criteria for the study, then oftentimes our response rates drop. So I'm very curious to see when we bring this into clinics, what does it look like? Does it hold up in the way that we expected it to with our randomized trials? And where does it land? So that's just a handful of ideas there.
Well, a bunch of very good ideas really brings tremendous hope that we could continue to improve treatment for people who need it. So, Joe, thank you again for the work that you've done, the work you're continuing to do, and for joining us today.
Thank you so much.
I also want to ask people to consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that if you donate a dollar for research, that dollar goes directly to the scientist. To make a gift, please visit us at the BBRF website or call 1-800-829-8289. This webinar has been recorded. If you missed any portion of the presentation or would like to share it with friends or family, please visit the events and webinars page on our website. And please join us again next month when we present another webinar. That will be on August 11th at 2 p.m. Eastern Time. We look forward to seeing you then. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Once again, thank you.